thank you for coming back. There's a housekeeping announcement. The Secretariat promised us that within half an hour, all reports will be available as a PDF. So let's see how it works. But in principle, the Secretariat aims to speed up, to have all reports of the various of the 4 discussion groups as a PDF file for you available. So thank you coming back. I hope you enjoyed this session 2, being part of the discussion group. At least I was involved in discussion group 2 and had a really fascinating day, and certainly our chair did a good job. So I think we reach some outcome. May I invite the first rapporteur, Matt Wright, to present the outcome of discussion group 1, more or less based on ALAP, so adverse outcome pathway and mechanisms of action. Matt, the floor is yours.
Okay, thank you. Good. Well, this is the first time I've acted as a conduit for the collective wisdom and thoughts of a group of around 25 people. It's been a very interesting experience. By last night, I was fairly comfortable. About 50 minutes ago, I started to have a panic attack. It's not so bad now. I've calmed down. So I have to say that it was an interesting discussion, but it really is what I'm about to show you are sort of collective, sort of average views. There are some disagreements, perhaps, or a difference in the way things should be emphasised. But given the time available, that's not too easy. Okay, so these were the questions that we were asked to address. I won't go through those right now. So question 1 was discuss the key epigenetic mechanisms and their potential relevance to key events under the MOA AOP analysis. So the kind of considerations and conclusions that we came up to was that we felt it was necessary to incorporate epigenetic changes to have a full understanding of the mechanisms of action. If it is an epigenetic toxin, of course, but certainly worth knowing if it isn't in any way, or the mechanism of epigenetics is not involved. Some of us, but not all of us, felt it was also important to accept that an adverse outcome isn't necessarily dependent on an epigenetic change. There are many cases where epigenetic changes don't have time to occur before an adverse outcome will occur. And we also felt that an epigenetic event could be a molecular initiating event, in other words, actually the cause, or a key event along the process or the mechanism of adverse outcome. Given the role of, let's say, methylation in changes in gene expression, it was thought that in almost all cases, epigenetic changes, if they occur, will also come with changes in gene expression, so changes in, let's say, messenger RNA expression, but also protein expression and so on. It was felt there was limited data that specific epigenetic changes lead to predictable adverse outcomes at this stage. And it is needed for causality in incorporating into the AOP approach. And we also considered that an MOA AOP framework would be helpful in contextualizing the epigenetic information. That's not one of my phrases, that's somebody else's. And we considered an epigenetic event or events may be a key event for transgenerational effects or delayed onset effects. So that concludes question 1. For question 2, we were asked to look at the in vitro and in vivo assays and non-testing strategies and so on available to generate the kind of data, and then consider the studies and study design and so on. And we decided not to answer that question because that was somewhat complicated and difficult. And we felt that we could pass it on to another group. Sorry about that. We felt that it's currently possible, undoubtedly, to detect epigenetic changes, but I think it should be acknowledged, certainly from an academic's point of view, that it's expensive still, although the contract companies available that significantly cut the price. But also it's still a niche skill, so it's not something that most labs can do still. They would need to bring in or pay for relevant expertise. And if you're going to eventually, let's say one day, use epigenetic data in risk assessment, then that will impact on the costs. So there will be a requirement for replicates, a time-dependent analysis, dose-dependent analysis in order for that data to eventually, potentially, lead to a point of departure determination. And that, of course, is going to be expensive. Second point there is adverse outcomes may involve transgenerational epigenetic changes. Human data, it was felt, only suggests a causative effect, but it's necessary, if it's necessary to demonstrate causality, and it is, this could be done in experimental models. Has that moved? Yeah. All right, sorry. It was felt that currently it's not possible to predict adverse effects from epigenetic changes. So if you detect epigenetic changes, you can't absolutely say that that's responsible for the adverse effect. And that is cognizant to specificity. And of course, from a regulatory point of view, there's validation that's required. So any model system for epigenetic analysis must be relevant and well characterized in the first place for the adverse outcome endpoint. In terms of methodology, members of the group made us aware of one publication on in vitro methods and 2 recent EquiTox reports on epigenetics and risk assessment. And they also include methodological aspects and very recent documents too. And there's also some other literature which we will probably add to the slide or the information if we can. In terms of in vitro methods, there are known limitations in any case to in vitro methods. With regard to epigenetics, what we've learned today is that there's aberrant epigenome maintenance, and that must complicate or have implications on any interpretations that anyone might use using these systems for risk assessment and so on. It was mentioned by members of the group that there may be a requirement for proliferating cells for these studies, and if metabolism might be important in the mechanism, then it might be necessary to provide that in some way, shape or form, however that may be. Question 3 was, how can epigenomics data be integrated with other omics data for MOA analysis and or AOP development? And we certainly felt that linking epigenomic data with other omics data would certainly be advantageous. It could be confirmatory about the mechanism. Actually, it could be just as useful to exclude mechanisms if there's no correlation and so on. So in that way, it enriches the MOA AOP database and is informative on the mechanism of action. And using the MOA AOP approach in a structured way will help to evaluate the information, such as epigenomics and so on. Has that moved? It hasn't. And so finally, question 4, there's quite a lot of discussion here on what are the data gaps and research needs. One of the first things that came up was reversibility of effects. If it's reversible, then obviously that would be something that would be needs more data, and therefore there could be a threshold effect, and that's going to require an appropriately powered set of studies with time and dose dependent data and so on. It's felt that an examination of species differences was essential for using animal models, for example, for regulatory purposes. So what changes or what effects do you see in species, and how relevant can that be to man? And on the same issue, in a way, in which in vitro models could you use and are relevant to the in vivo situation, given what was said earlier? And it was mentioned also in the talks and by members of the group that there is a normal human variation in the human epigenome. So to what extent is that variability an impact? And that was felt that more data is needed. In terms of those sorts of things, and also clinical data, there is apparently data coming out from clinical groups on epigenome signatures from disease organs, and that may be, and also perhaps epigenetic biomarkers, and that could be very useful for our purposes. It also would be helpful to define any of the epigenetic mechanisms in more detail in chronic diseases, such as developmental origins of health and disease. It was felt that it was unlikely that you're going to get a sort of major epigenetic markers, if you like, for every chemical insult, but there may be a possibility for different classes of chemicals to have sort of signature epigenetic changes or epigenetic biomarkers. It was felt there was limited data on epigenetic changes after different chemical exposures as key events. They felt that more data was required to see if these changes are specific key events for the classes of chemicals, and that really is related to the previous point. And again, what specific epigenetic changes lead to predictable adverse outcomes? There's little information available. What specific epigenetic biomarkers are also predictive of these adverse outcomes? And can we ever say something is epigenotoxic? They felt that we need to consider other disciplines in order to, such as clinicians and so on, in order to sort of answer some of these questions. And again, for our use, validation is essential, and in many ways, a lot of that work needs to be done. So in that way, which epigenetic changes are causative for adverse effects, and it's felt that epigenetics can potentially play a role in all non-communicable diseases. It may not be essential, but it may play a role. In terms of in vitro and in vivo assays, it's felt more mechanistic data and understanding is required. Perhaps somewhat tongue-in-cheek, a quick screening assay for identification of potential epigenotoxins would be very nice. So something like an Ames test or something like that, very quickly. That's a long way in the future, I'm sure. And to make use also of the current human epidemiological and clinical data. And that's it.
Thank you, Matt. For this comprehensive summary of your discussion group. For sure it might be a major task during night, or perhaps also this morning, but thank you. But I would, because there are still a few minutes left for discussion group 1, so I would like to open this paper first, giving the chance of the other participants of discussion group 1 to add information, what perhaps was not in the presentation. And if there are no questions or additions, then I would like to open the paper for general discussion. So who is... Professor Ghali.
I think that you touched on one of the first slides, one important point, at least in my mind. Your question was if it might be a molecular initiating event or a key event. That is what I believe, because I still think that we are focusing too much on the epigenetic change per se. But we have to consider that is kind of phenomenon among many others. So why we are just putting so much attention on that point and not we consider that in the risk assessment, this is one or key event or a molecular event, as you said. That's, I mean, my opinion. And I like very much what you presented. I like very much because we did a similar presentation. Thank you.
I guess there's no response to that, really. Thank you very much.
So, if there are no other comments, remarks, then I would say thank Matt, thank the special guest. May I invite Raffaella Corfi to present the outcome of discussion group 2.
So as the previous rapporteur said, it's quite challenging to capture the discussion, and especially in our group we had a very lively discussion. So I will try to summarize the major points that were made, but also would like to give you also some feeling on the dynamics of our discussion. So you will see that some points recurred more times. So this was our group, and thanks to the excellent chair, and we had also co-rapporteur Marco. So we were given 4 questions, very challenging questions. And, well, these are the questions, and actually, yes, it's okay. So the question for our breakout group assumed we thought more aspirational readiness for the science, but the presentation raised more questions. So maybe I just go back here, and actually we were quite happy because we started our discussion with some consensus, and the consensus was among the participants that the presentation we heard yesterday were excellent, and also they really presented the compelling science. And there is evidence that there are epigenetic changes in the genome that may or not play a role in adverse effect. So the first questions we had to ask, although I have to say we were tempted to jump to question number 4 initially, but we worked diligently through the question in the right order. So, do toxicity studies with their current design allow to measure adverse effects resulting from epigenetic changes? And in our discussion, epigenetic changes were considered in a general manner, so we didn't focus on the different types of epigenetic changes. We felt this was not necessary in our discussion. So the response was yes, toxicity study may, in principle, allow to monitor changes and measure adverse effect resulting from epigenetic changes. However, current knowledge does not allow to agree on which endpoints to capture and on whether those represent an adverse outcome or a step in the pathway or mechanism. Then also, I mean, this is a more general question was raised, is can our current risk assessment paradigm capture the biological complexity? And it was recognized that there are limitations, for example, in the assessment of multiple exposure, in timing or the window of exposure, and multi-generational and transgenerational effects. Here I reported two quotes from members of the group. Biology is more complex than what our current tox model represents. And then an interesting one was, it was asked, is the paradigm ready for the science, or is the science ready for the paradigm? And maybe there is a disconnection between the two. So in the risk assessment framework, so we were convinced that epigenetic data could help to reveal AOPs, and we heard in the previous presentation that this is the case, and you're representing key events. So we were not so brave to say that it may represent molecular initial events, but we didn't go so much in depth into this discussion. It may be also helpful to identify markers, early markers of effects, which may be more sensitive. However, there was concern that too high sensitivity may become irrelevant. And also, these good epigenetic changes could help explain inter-individual variability. However, if we don't know the baseline and the contribution of background, we can only see changes at the individual level. And so it was asked, are these main regulators or effects of modifiers? Are we expected to run personalized risk assessment, as it is now happening in medicine? Should we consider epigenetic changes as baseline effects in the population? So also it was emphasized that we are working for the public, and more complex risk assessment may not necessarily be beneficial. We need to be able to disseminate the message in a simple way. So there was some discussion, and not everybody agreed on that. I mean, the agreement was that the message should be simple, but also it can be given based on a complex science, and it's important not to compromise science. So there is a need for better understand normal and background biology in order to appreciate epigenetic effects. What is normal also? The challenge is due to the fact that we have several polymorphisms. Also, it was recognized that if we add toxicity parameters, this may have an added value. But also there was concern that this would add burden to the toxicology toxicity testing and may generate high complexity as well as too many spurious changes and in the end increased uncertainty. So then we went on to question number 2. Well, you can read the question. Discuss key issues to integrate in vivo data reporting epigenetic effects, including non-coding RNA-based biomarker, in BMD modeling, or identification of reference endpoints for hazard characterization. So you have heard what we responded for question 1. And we thought maybe this question, was a bit provocative or posed in a very optimistic way. And the group felt that integration is not the issue yet, let's say. But understanding dependency or the association between epigenetic changes and adversity, as we heard in the previous presentation. However, also it was said that even when those data or additional data, not necessarily epigenetic data, are available, they are sometimes not integrated into risk assessment for different reasons. For example, sometimes for policy reasons, lack of experience, and difficulty in interpretation. And this might be the case, for example, for omics data still. So we talked about adversity, so we thought it was important to go back and to look at the definition used by EFSA on what is an adverse effect. An effect is considered adverse when leading to change in the morphology, physiology, growth, development, production, reproduction, or lifespan of an organism, system, or subpopulation that results in an impairment of functional capacity to compensate for additional stress or an increase in susceptibility to other influences. We also looked at the biological relevant effect definition used by EFSA. But there was agreement that based on what we heard yesterday morning, and I think we agree also with Group 1 here, it was difficult to understand or to derive what is an adverse effect of epigenetic changes. So important is first to define the adverse effect, both in relation to the different endpoints, but also versus modulation of underlying biological variability. And of course, once these are defined, there are test methods available. We then, as we already heard, needs to be optimized, validated, and maybe lead also to test guideline that can be used in a harmonized way. So, if sufficient data on epigenetic changes and a clear sequence of events from early changes to adverse effects, if there are sufficient data on epigenetic changes and there is understanding on the sequence of events from early to adverse effect, then hazard characterization would be feasible. Epigenetic changes may have an impact on hazard characterization. We also thought that there was a need to rethink the measure of adverse effects in the current risk assessment paradigm. So this was a more general question, but it came out in our discussion several times. Can animal models mimic appropriately human effect? And maybe epidemiology can help identify more relevant and global endpoints of adversity. We also continued with quite robust discussion on pragmatic approach to risk assessments versus a more aspirational risk assessment. So we had really different views in the room. And so one of the questions was, can the current risk assessment framework accommodate all the emerging science? And also was said that every study may be or is a compromise between design and choices. Then we went to question 3, was what in vivo, in vitro assays and non-testing strategies are available to generate relevant data on epigenetic changes for incorporation into risk assessment frameworks. And here we had here the term non-testing strategies, so that was something new that was not covered in the previous questions. And we thought that in silico models could be useful if we would have data on more chemical, which we don't have at the moment, as part of a holistic approach to set priority. So I was asked to read this sentence. And we also thought that maybe in the future, QSR could be based or built on what on data regarding methylation. There is a need to rely more on well-designed epidemiological study that measure epigenetics and the exposome, or understanding the exposome is important. Again, a lot of debate, again, on what is important for a risk assessor. And some people in the room thought that ultimately the goal is to establish a point of departure. So maybe we should focus on the question, can we rely on epigenetic derived point of departures? Of course, if provided we have the data are available, as opposed to maybe majority of the members of the group thought that the risk assessment framework is an overall assessment as embraced by the 21st century approach that points to the integration of information. So also, it was mentioned that current assays maybe already capture epigenetic effects within the uncertainty of the risk assessment, at least for cancer endpoints. And also, there is some expectation that there is a need to address transgenerational effects. So does the current risk assessment model capture them? And maybe this is, as said earlier, epigenetic can help. So then we move to our last question. What are the data gap and the research needs? First, we thought it is really fundamental to ask the right questions. There are some basic questions that need to be answered first. Okay, we now know that epigenetic modifications do occur. How this modification modulates gene expression. Then sequentially, once we can answer the question, we can ask following question: is the sequence, if the consequence are adverse effects, can we understand the extent to which these are adverse? And if we understand the phenomena, can we integrate it in risk assessment? Can we integrate them in risk assessment? Which endpoints are regarded of specific importance? So, it was clear that, I think also said before we can explain how to use epigenetic in risk assessment, we need to better understand the baseline biology and variability, including interspecies difference. So mechanistic understanding of epigenetic changes and their association to gene expression, and that's why we were really, really interested in the outcome of the first group. The need to determine association between early events and adverse outcome and the sequence of events. This was said already. And also an important question is, in the future, when we know more, should we add an endpoint that is not necessarily adverse to our risk assessment framework? Most of the publication focus on few chemicals at high exposure and also on few biological parameters, and there's clearly a need to extend our knowledge with information on more chemicals and to identify characteristic patterns for specific endpoints. So how can we generalize what we know now? Need for epidemiological studies, but with relevant biomarker of exposure and epigenetic effects that are integrated with data from appropriate tox studies. there is a need for more interaction, we thought, between toxicologists and epidemiologists. Some jokes were made here when we discussed this point. And I think as the first group, we can say that translational science is needed. We have heard that this is already ongoing in different countries with more or less success. So I think this was my last slide. I thank all the participants for the discussions, and thank you for your attention.
Thank you. Again, an excellent summary. Took a lot of time to do, so I can imagine that this really gives you some pressure together with the share and co-shares. Again, the paper is open for questions because we have plenty of time to... Oh, excuse me.
Yes, well that's very interesting. And in working group 4, we have discussed a number of points you brought, you presented so well here. I really liked your point on the question on whether non-adverse epigenetic endpoints should be included. And thinking about it, that might actually be interesting. Because at regions where epigenetic changes can occur, but there are no phenotypic consequences, maybe those changes will become bigger and can be therefore be more readily detected. So I think that's quite interesting. So maybe certain retrotransposon elements, for instance, are highly susceptible to methylation changes, for instance, but that will not necessarily have a detrimental effect on phenotype. But that could be maybe more readily be monitored than a change at a transcription factor gene involved in development, where even slight changes will immediately lead to rather traumatic phenotypes. So I thought that was very interesting. I have 2 other little points which I think we brought up in group 4. So we also wondered whether microRNA should be considered epigenetics, and should those be included in the umbrella of epigenetics. And maybe we'll come back to that later, but we thought that possibly microRNAs are maybe not epigenetic. But that's
we didn't touch on this point, no.
But I noticed you had a microRNA on one of your...
in the question, not in the response.
And another point we discussed is whether effects that seem to be passed on to next generation should be called intragenerational or intergenerational. So some of us thought that maybe intergenerational would be better because there's some sort of a link between the exposure in one generation to the phenotypic effect seen in the next. Thank you.
Thank you.
Tina, you?
Yeah, I'm just wondering if Jean-Michel, if he wants to talk about his point. He's not looking at me, about the changes epigenetics might have to baseline population that might lead to future susceptibility. So not an immediately observed adverse effect, but epigenetic changes that make changes to the population that result in future susceptibility.
Jean-Michel, you ready?
Yes, but the question I raised within this working group actually has been dealt with in the working group number 1. And I think it has been well addressed and well presented by Matt in the previous, because here my impression was we focused more on the impact, the possible impact of epigenetic changes on the toxicity of a contaminant. And I was wondering whether we should also focus, for example, on the role of other compounds, such as nutrition, on the susceptibility of individuals to be more or less, or to develop more or less adverse effect to further exposure to contaminant. And actually, this has been addressed by the breakout group number 1, And I was very happy to listen your explanation, because I think both reflection of number 1 and number 2 are very complementary.
Thank you. There's another question.
Well, it's not a question. It's more a comment. First of all, I like very much what you said, bringing emerging science into risk assessment. I just, with a small caveat, we should try to avoid to drown into complexity. I mean, if we are working on models and assessment, we have to sort of reduce, make an effort to reduce complexity. This is really in the meaning of a model. I think there are two main fields of adversity that are emerging now, and one was just mentioned is enhancing susceptibility to further environmental, let's say, insults. The other one is the true transgeneration effects, which is, an effect induced an exposure in generation 1 without any true mutagenic effects, interaction with DNA, but which is transferred to the next generations. To me, these are 2 points where toxicologists or future generations, of course, should work.
Yes, and I think our group completely agreed with this point, yes.
This was well treated, but of course, yes, it's noted. So, nobody else who wants to question us what we did in discussion group 2? So then again, Raffaella, thank you. So may I invite the third rapporteur of discussion group 3, Erik Nielsen. Erik, the floor is yours. There's plenty of time, so.
Thank you. And in preparing this presentation, I had been concerned about time available. So after having after preparing a report of what we had talked about for the group this morning, I actually condensed it and made it smaller. We may have lots of time. So, to make everybody understand what we talked about, we emphasized how epigenetic changes are a normal mechanism of responding to environment. We wanted to point out and emphasize that, epigenetic changes induced by environmental conditions can certainly, as the other groups have said, have long-term physiological effects, and also for farmed animals. These may be transgenerationally inherited, but we don't have very much information on that in farmed animals yet. So from the standpoint of considering farmed animals and risk assessment, you can look at risk assessment in several different areas. You can have risk assessment of food safety of the food from farmed animals for humans. You can have risk assessment for the farmed animals themselves. Do they have increased risks of disease or increased risk of later poor welfare that can be associated with epigenetic change, induced epigenetic change. And there can be risk assessment that speaks to whether there is a risk to the food supply. This would be a food security issue. And certainly these are all valid risk assessment uses, but they're certainly not the same as each other. Also, we wanted to speak on, oops, I missed one. Also, we wanted to speak on what we would call the special case of cloned animals, or those that are involved or derived from assisted reproductive technologies. And the reason that we discussed this topic is that there was a call, a desire within EFSA to speak more on this. So before we get into our specific questions, I thought I would present the results of our discussions, and that is that the evidence suggests that assisted reproductive technologies can result in epigenetic changes. From a food safety standpoint, animals derived from reproductive technologies and cloned animals are as safe to consume as other animals. Certainly, if these animals have increased risks of certain kinds of disease later in life, from the food safety aspect, that is very likely dealt with by the normal current practices of evaluating the safety of farmed foods. Epigenetic data, therefore, is not expected to replace current food safety practices, although in the future it may add to them. Also, there's evidence from the research that the increased disease rates currently seen in animals derived from artificial reproductive technologies, those increased disease instances may actually reduce or may be improved with technical refinements for the reproductive technologies themselves. The examples had to do with overlarge calves derived from in vitro cultured embryos, and apparently if you work to change culture conditions, these problems decrease. But to go on to our specific questions, one of them was, can changes in epigenetic marks lead to predictable phenotypic changes in farmed animals? And for what limited data there is in the field, sometimes yes, sometimes no, but mostly the opinion was that the evidence is very limited for farm species. Please support research in this area. There is a correlation between epigenetic changes and phenotype, but it's often correlative data. There's not often evidence of causal links, and that's been referred to in our previous presenters also. So our second question had to do with what are key epigenetic marks if there are epigenetic changes that are induced by factors in the environment. Are certain of them key? Are changes in these epigenetic marks biologically significant? How do you define normal? Again, issues that have been also addressed by our previous presenters. We decided that there may actually not be specific identifiable key epigenetic marks or biomarkers, but it's more likely that there may be groups or suites of epigenetic changes that may lead you to think that a phenotypic change is likely, which is statistically true and not as, but harder to understand. And we also spoke to the issue of not knowing what normal epigenetic variation is within farmed species. How that relates to risk assessment may have to do with then trying to decide what we consider to be a normal variation in epigenetic of the epigenome. From a risk assessment or from a regulatory standpoint, it may be easier to identify the absence of certain defined hazards or diseases than it would be to try to define what is normal. That's not as satisfying, but people can more clearly come to an agreement. And as far as whether or not epigenetic changes are biologically significant, again, from a risk assessment standpoint of the food from farmed animals, are the food products from farmed animals with epigenetic changes safe for humans to eat? Likely yes. Similar as what you would see for cloned animals or things like that. There's not sufficient evidence yet that adding epigenetic data or epigenetic tools will improve food safety risk assessment. So for that specific kind of risk assessment, the addition of epigenetic data, there's no indication that food safety would be improved. And finally, are there food supply security concerns associated with farm animals having epigenetic changes? And the answer could be yes. If epigenetic changes are associated with a phenotypic change of increases in disease, decreases in reproduction, shortening of lifespan, then that brings into, that speaks to the kind of risk assessment that goes with food security. And in this case, certainly epigenetic data may be very important. And it is also related to this case where we would perhaps have to start looking at the chance of future generations of farmed animals having inherited epigenetic transgenerational changes in phenotype, and trying to then predict, does that have an implication for the food supply? Oops. And question 3. Yes. Okay. And question 3 was, what are the available methods for assaying epigenetic marks? What would be necessary to validate them? Again, other groups have spoken to this, perhaps in more depth than we. But from the standpoint of talking about farmed animals, the best developed assays of the epigenome are for DNA methylation. For everything else, histone modifications, non-coding RNAs that are not sequence specific, changes in chromatin structure, all of those other things that come under the umbrella of epigenetic changes that affect gene expression, the techniques and the assays to evaluate those in farmed animals are less developed, sometimes non-existent. And that's a big area of need for future research. Now, from a risk assessment standpoint, validation is an important concept. Validation of marks as being in a causal pathway would be nice to know, but may not be something that one needs to know. And on this point, opinions were varied within the group. Now, as far as thinking of validation of epigenetic changes as being informative for risk assessment of various kinds, one would want to know, is there a strong link to the phenotype? Are the assays that figure out epigenetic change, are they robust and repeatable? Assays may have to be worked out differently for cells from different species. Do the effects of certain epigenetic changes hold across tissues or across species or across breeds as far as the expected phenotypic change outcome goes? And then we have the knowledge gaps. And of course, in the case of farmed animals, there are many knowledge gaps about epigenetic mechanisms. That's really no different than what anybody else in other groups has been speaking of. There is certainly a large need for descriptive research, basic research in farmed animals. Much less is known than for humans and rodents, Drosophila. And in some cases, some even basic physiological information about the normal windows of epigenetic reprogramming of germ cells, for example, in chickens, is not well known, and even less so from some of the other farm species. We need to put more information or more research into the potential of and reversibility of epigenetic changes, technical tools for research in farmed animals, such as more complete databases, even well-annotated genomes, cell isolation techniques for different species, bioinformatic pipelines if they happen to differ from rodent models are often not present. And we thought that the mitochondrial DNA epigenome is an understudied area because it often is, changes there can lead to metabolic defects, which do occur as, reliably in farmed animal species. As far as knowledge gaps go, one goal would be to eventually be able to identify regions of the genome where epigenetic changes are linked to phenotypic change. We would all love that, maybe in the future. Specifically for risk assessment in food safety, food security, it will be necessary to do studies with each group of farmed animals in order to get information relative to those groups. So we may have to actually do epigenetic research in Holsteins versus Guernsey dairy cattle in order to know if the changes, epigenetic changes, are linked to phenotype differently in different breeds. And again, that would have implications for risk assessment. Let's see, multi-generational studies or transgenerational studies are an important aspect, especially from the standpoint of risk assessment for food security. Of course, if you're speaking of animals with fairly long lifespans, these are very expensive studies, but still, they should be done. Studies of epigenetic changes in the germline might be something one can do in the near term, sort of as a proxy for transgenerational effects. And this is the end of our shortened presentation. I am happy to answer any questions that you might have.
Thank you, Eric. Again, a fantastic job you did, really compliments. The paper is open for questions. You are willing to?
Yes, certainly.
Of course.
Sorry for taking again the floor. I think your group, the discussion group 3, just gut feeling at an excellent point, making the issue, evidencing the issue of mitochondrial DNA epigenome. Do you think it's relevant also in the field of mammalian toxicologies or something to be explored? Or I'm just asking the assembly.
Yeah, within our group we had a person who was particularly interested in this area of research. Is he here? If so, he can answer your question. Go ahead, press the button. He's shaking his head. Okay. No.
Okay, thanks.
Okay.
Gianfranco Brambilla, I am from the same institute of my colleague Alberto, so sometimes there are some connection problems or whatever. Well, we know that some methylation of mitochondrial DNA is related to some heart failure and muscle diseases like rhabdomyolysis and whatever. In high inbred pigs, we have a very nice degenerative disease called Malbury heart disease. And basically, this is based on mitochondrial failure. And of course, the adverse outcome pathway is driven by reactive oxygen species and their metabolites.
I think this is a very interesting question, but it can also be argued that in the field people discuss on whether there is any mitochondrial DNA methylation. This is a very active discussion, and I have been in contact with groups who seem to be working rather seriously, who bluntly say there isn't any. This is all, so I think this is then in these kind of general texts, one has to be careful with putting something on mitochondrial DNA methylation.
We may nevertheless take this risk.
Yes, I think one needs to be, it is highly disputed.
Yes, okay.
Take the floor.
At the bottom of the button.
I have a comment on the introductory part. I think you seem to suggest that there is a higher incidence of diseases on ARTs. In general, this is simply not true. If we have concurrent data for, let's say, artificial insemination for embryo transfer for 25 years, there is nothing. IVP, there was some concern when we had this oversized calf, which is gone. So it's a bit related to the oversized offspring initially from cloning, but there's not a generally higher susceptibility to diseases. So I mean, in that sense, is simply not correct.
And certainly, we would be happy to adjust the language in our reports.
That word disease is the wrong word.
I see.
Could you use the mic?
Lowest button.
Oh, sorry. Yeah, the word disease should not be on that slide.
Okay, we can do that.
Thank you.
Yes.
Yeah, in line with that, I was also wondering, in addition to consequences on food safety and food security, it would also be interesting to see the consequences of animal welfare. And that is one thing, the risk of increased weight of calves, birth weight of calves, for example, that maybe it should be mentioned.
And in fact, we did make a point that risk assessment for the welfare or increased risk of disease of the farmed animals is an important thing to explore, and maybe one of those areas that really is helped by the addition of epigenetic data.
I have an additional comment. I think we discussed it this morning, and I think, but it's important to have it in that text. The thing what I think we have agreed upon is that these epigenetic data might provide additional information with regard to food safety. That means the sentence that we put in now is that likely not improve food safety assessment. I would eliminate it. I said it provides additional data that might help to improve food safety assessment. That's for sure. That's what we're discussing. It's an additional tool in the toolbox, and we have additional tools to improve our assessment, and that's what should be in there.
And I would certainly agree that more data is never a problem. I think we also had considered that at our current level of knowledge, the addition of epigenetic data to our food safety assessment may not add additional safety. But in the future, possibly so.
We as scientists are looking to the future.
But for sure, as we promised, as we start drafting also your work, it will send around. So please find some kind of compromise. Otherwise, we have different opinions. There's another question, please. In the back.
It's not so much a question, perhaps a quick way to resolve some of this discussion. The word may means different things to different people. So I think may means might in the course of these discussions, not will or likely will. So perhaps that's just an easy way for us to edit the slide. I was part of this discussion group as well. So I think it's important to get the appropriate message across. These things might provide additional information. It's not that they will show different things. They might.
Thanks. There are also native speaking Englishmen in the audience, so thank you for helping us. Please.
Yeah. I also want to make a point about the techniques, because I think this is especially relevant for farm animals, that we need more genome-wide, cheap, unbiased techniques to address biomarkers in large populations, which is especially required for farm animals.
And I would agree.
Just a point of clarification. You indicated there were divergent opinions on the issue of validation, and I wondered if you could give us some flavour of what the concerns were. I mean, I would understand that validation is for a purpose. I mean, if you just do a normal experiment in a laboratory, you don't. The validation is something you do in your experiment. Validation for a regulatory purpose might be a completely different thing. But I mean, I don't want to prejudge your.
And I may not be the right person to ask this question because my impression from the study was exactly your point, that the term validation in the context of risk assessment might have a specific meaning. Myself being an academic researcher, I'm not familiar with that. But others who are more involved in regulation and risk assessment, my impression was that validation in respect to risk assessment does have a specific purpose and a specific meaning that might be different than my assays. And so we tried to put in some language about the concerns about that.
Reinhilde?
Yeah, I think indeed we mix two things, the validation of the method itself in the technical manner of more than one lab tested and so on. And we mixed that a bit with the difference in opinion of the causality that such a method on biomarkers can offer us. And that's where we were unclear whether or not we need such methods to inform us about the mechanism behind and the causality that you are looking at when doing risk assessment.
And that would be causality linking epigenetic changes in farm animals to adverse outcomes or something like that. Or to any phenotypic change.
Caused by the stressor on their assessments.
Okay.
Yeah.
Thank you. Thank you, Eric. Thank you, discussion group 3. May I now invite David to present a summary of all the discussions convened in discussion group 4. David, the floor is yours.
Is it working? Yeah, good, brilliant. Thank you. I think I've. Okay, yeah. So I think I've got in total an hour and 10 minutes to present. So I'm looking forward to filling the whole of that time to fully summarise our discussions and give you some of my life story.
I'm not joking. I'm not joking because we need some time, otherwise we have an order proposition because we have also to draft take-home message and that takes a little bit of time. So take your time.
Okay, so here goes. Now we had the smallest group, and because of that we actually had, I think, a lot of focus and a lot of consensus and very few arguments actually for a discussion group. I think that's a fair statement to make. So I think what we present is a reasonably consensual view of the four questions that we addressed, which is, I know, can be unusual for these kind of meetings. So that was all jolly cheerful, really. Let's get this thing working. Oh, there we go. Come on, go back one. No.
Some assistance?
Oh, here we go. Right. I'm going to take my time. Right. Okay. The first thing that we started off with was, before we got to our questions, we had a really fundamental discussion. We were focusing on environmental risk assessment. So we had mainly environmental risk ecotoxicologists. We also had some experts who'd worked on human epigenetics and more widely on just epigenetic mechanisms across organisms, and that was really useful to have that mix of people. But we started off really in thinking about environmental risk assessment on the kind of fundamental question of why care? Is epigenetics going to be relevant at all in environmental and ecological risk assessment? So that was really our first question that we discussed, was why care? We identified things that other groups have identified. We identified that chemicals can affect the epigenome, that they can drive adaptive change. Epigenetic mechanisms may be involved in adaptive changes. They might be beneficial or detrimental, and that might depend on which organism is living in what kind of environment. We also identified that epigenetic dysregulation can cause adverse phenotypes, so it can be doing something important. It may explain particular modes of action and adverse outcomes. Some of those were taken from human toxicology around the nature of carcinogenesis. And we also discussed how important that kind of mechanistic understanding was in ecological risk assessment when we're thinking about the health of populations, not necessarily about the health of any given individual. Although we did also recognise that currently there's an increasing focus in ecological risk assessment on the individual, both from a modelling perspective and also in sort of respect to the protection of endangered species. So it's possibly no longer the case that ecotoxicologists only think about populations. They're starting to think about the individuals that make up those populations, and certainly when those individuals are nice and big and fluffy and or pretty, they're beginning to care about individuals even more, and particularly when they're rare. Come on. Yep. So these were our 4 questions. They were really about they were really centered on what current tests tell us, what new kind of epigenetic understanding we might want to include in current ecological risk assessment, what kind of methods we might want to use to get that knowledge, and what the data and research gaps are to deliver that knowledge so that we can begin to use such methods should they be deemed appropriate from what we begin to understand and know. So first, dealing with current state of the art. That was the question, and it was good because we had the person who wrote the questions in the meeting, so we could really harass him about the quality of the questions. Wherever he is. He's sitting at the back in the corner. The first one was about, do ecotoxicological laboratory studies with their current design allow measurement of adverse effects resulting from epigenetic change? We were asked to give examples. We weren't always good at giving examples, but we tried. Yeah, come on. It's going to flip again. Right, the first, so question one, that we identified that the tests that we were using measure effects that could be influenced by epigenetic mechanisms, but the current things we're measuring in ecotoxicology are apical endpoints, so we're measuring survival, growth and reproduction. That's because the focus in ecotoxicology has been on protecting populations and its effects on those endpoints that contribute to the long-term health and survival of populations. So it's difficult to ascribe causality of observed effect from current tests. That relates to knowledge about mode of action, but generally, but about epigenetic mechanisms and their involvement also. We said that we identified that there are research studies in ecotoxicology starting to go beyond standard tests to start to attribute changes to epigenetic effects. So there's looking at adaptive mechanism, there's work on adaptive mechanism, so that's looking at epigenetic change within generations, and there's also intergenerational experiments starting to take place in ecotoxicology. There are certainly, this kind of area is strong among some fields, like radioecology really seems to have taken up this idea of long-term and multi-generational, intergenerational studies. And it's something that's starting to move into other areas of ecotoxicology as well. we recognise that in ecotoxicology we're often working with species that are easy to look at and easy to culture and easy to cultivate. So we're often working on clonal organisms, and it's true that a lot of the intergenerational studies that have been done so far have been on either Daphnia or C. elegans, because you can, firstly, they have fast generation times and they're also clonal in nature. So they will give us some information about the relative contribution of epigenetic changes versus genetic variability in populations. And in some of these species, you can also match those studies in inbred lines with outbred strains as well. So we can look at the different contributions. So there's important information we're going to be able to gain from the kind of test organisms we work with and we know how to work with. We recognise there are already two standardised two-generational tests in a marine. One in a marine copiapod, which I don't think we ever were able to remember what it actually was, but it was a marine copiapod, and one in Medaka, which I can't remember the Latin name for, and they've been ring tested and standardised. There's also a two-generational test for Daphnia that's in development at the moment, so that's the pre-ring test results of that have been published. There's a limited information though, and I noticed this came up for farm animals well. We just lack fundamental information on epigenetic mechanisms in a lot of taxa. That includes in even major ecotoxicological models. We don't know anything about things, we know relatively little about things like Daphnia, and even less when it comes to things like earthworms and springtails. Things I know about are used as in ecotoxicology as standard test organisms. So what we're in a position is it's difficult to gauge how suitable those models are for predicting effects on species that we might be wanting to protect, because we don't know the fundamental epigenetics of the species we're working with in the lab. We certainly don't know much about the species we want to protect in the environment. So we identified, of course, the idea that there was persistence of effects after stress or exposure. We also identify that current tests don't include that pre-exposure assessment phase. Current protocols involve short-term exposure, measuring effect, and that's the end. They don't look at post-exposure monitoring, so we're not able to understand that memory effect, whether it's there or not. Some ecological tests don't look at all life stages. They look at certain life stages, so we might miss out on looking at the windows of susceptibility. That was a general area that we thought knowledge of was missing. And we also recognised that because we had people talking about both epidemiological studies and also laboratory studies, and recognised that there wasn't much linking of those two areas actually. So we are in a position, we certainly think there's benefit to both, and there's probably benefit in linking the two more explicitly. And studies that try to look at epidemiological effects and link those to laboratory studies that are mechanistic, or take mechanistic studies from the laboratory and find representative field experiments where we can look for those effects in the field and whether they're relevant in nature, are probably going to be quite powerful things to do. And particularly, in ecotoxicology, we tend to go, because we can work with organisms in the lab, we tend to focus more on the lab than the field. So the epidemiological element is largely missing, I think, or we thought. Our second question was about integrating in data and biomarkers and mechanisms into risk assessment. So what were the key issues that might sort of facilitate or present this kind of integration? The first that came up was regulatory acceptance. We recognise that currently we don't need to do this, so at the moment that's a barrier to the development of methods. We also recognise that data standards were missing. The transcriptomic community had already been involved in development of data standards, and there were other areas where that had been done. That work was yet to be done for work on mechanistic understanding in epigenetics. We thought there was issues around what experimental designs were suitable and how we would work, and the problem about mapping mode of action and adverse phenotype together, the fact that we needed to do those two things, we needed to understand the epigenome and the adverse phenotype and make the link between the two, that was still missing. We didn't know a lot about size of effects that we needed to worry about. We don't know if, we mentioned we don't know a lot about the mechanisms in organisms, so if we don't know much about the mechanisms, it means that we don't know much about the baseline. If we don't know much about the baseline, it means we don't know much about what adverse looks like and how much deviation from baseline we need to worry about. How long, how many, what loci are affected, what's causing the effect, etc. We also discussed the relative value of genome-wide versus loci-specific. We needed to know more about what loci we looked at. Global methods for DNA methylation were easy to do, but how powerful were they? How quickly did we need to move to loci-specific analysis? We could only do that if we needed to know what loci to look at, and we had an interplay between costs and benefits. There's no point spending all the time, lots of time looking at things if we can't interpret them. So we missed that kind of fundamental information. And we also discussed this idea about the epigenome being valuable as a surrogate for adversity. So one of the problems we have in the environment is around pulsed exposure or pulsed exposures, and knowing and biomarkers and finding biomarkers of exposure that is no longer relevant. We have pulsed exposures a lot in ecosystems. Can the epigenome act as something that retains a memory of that pulse exposure, so we don't have to be there when the exposure happens, but we're able to know that that population is subject to pulse stress? That's very relevant in a lot of, particularly things like river systems as well, where the exposure might be only for a short time, but might be quite comprehensive. And it's, there are certainly questions about the value of chemical monitoring in rivers. But can we use biological monitoring in the epigenome because it keeps a memory of that exposure? That's just one example. Come on. This is the slowest thing ever. It's really frustrating. Right, so measurement endpoints. So first thing we recognise is there are certain macrophenotypes you can measure that tell us that there's been an epigenetic change. I can't remember how the subject of big blue rats came up, but it did come up. But we also talked about yellow tomatoes and so on. We also talked about turtle sex ratios. So actually measuring macrophenotypes that we know are dependent on epigenetic change, we shouldn't forget to do that. Just because we can go in and do other things, we shouldn't remember that we need to measure phenotypic effects when they're there. DNA methylation, it was also mentioned in the farm animal talk as the first place to start. There's plenty of examples of a role in inheritance. We need to look at also methods that can, we now understand this life cycle of methylation. So we also need methods that can look both at DNA methylation and hydroxymethylation and know what techniques we're looking at, what's measuring what, and so on. Analysis of histones was also relevant. We recognised that this was valuable in ecotoxicology. There's a high evolutionary conservation of histone modifications. Unlike DNA methylation, which seems to vary in its role between species, there seems to be the phrase from fungi to man was mentioned, and whenever you're looking from fungi to man, you're covering an awful lot of diversity. So, that's certainly important. I hope I captured that point correctly. We discussed this idea about microRNAs as mechanisms for transgenerational inheritance, and this is where our discussion about whether microRNAs were really part of the epigenome or the transcriptome. We also discussed that there was methylation of the loci encoding microRNAs, so there was an interplay between the two. We mentioned about histone acetylation being a dynamic, and some long-coding RNAs also being a dynamic part of this transcriptome. So we were left really with focusing on DNA methylation and histone methylation as the major places to start looking at where we needed to work. So then we went on to look at the tools that were out there for different organisms and discussing the molecular and bioinformatic tools, databases and models. So this could have been a really long discussion about the techniques of different ways of looking at DNA methylation and looking at different next generation sequencing techniques, and it could have been but it wasn't. And I think I was quite glad of that actually. We actually focused on more the general, although there's certainly plenty of scope here to really talk about methods, and there's plenty of work that needs to be done about looking at the power of different methods. So we said there was a lot of need to understand the basic biology of systems, to understand chemical responses to the genome in its natural context. We're missing that kind of essential information. Different organisms are obvious choices. There's fundamental research on crop plant quality and from model organism studies that are providing us with new tools. So as people worrying about species living in the environment, we should also be very conscious of the work that's going on with the species that now seemingly only live in the laboratory. The C. elegans and the nematodes, and also the things like crop plants that are no longer really proper species anymore. In many respects, they're now been manipulated to be to an almost artificial form. There's lots of tools coming out, and we can use those. We also mentioned about where we start analysis. We used to, if we start, we have a question about, do we analyse at the organism, at the tissue, at cells? We discussed the possibility of looking at single-cell DNA methylation analysis and how powerful that was to focus really on specific adverse cell types where we knew that they were a target. And we said that there was also a need to understand variability of population phenotypes. For a species living in the environment, variability in the laboratory, variability can be an issue. For something living in the environment, variability is an important benefit. And just because we see a variable response doesn't mean that that's not a meaningful or important response for us to understand the potential a population has to adapt to a particular exposure. Right. So we noted there are some species databases where the epigenome and the genome are starting to be laid over each other. Main model organisms would be some of those. Daphnia is some of those. We've also been involved in doing stuff like that for earthworms as well. So there is beginning to be databases out there. There are established microRNA databases. There's great published resources on histone modifications because of this high conservation and the ability to transfer knowledge between systems. It also means that a lot of the tools that are out there designed to look at histone modifications, the antibodies for pull-down studies and so on, will work between different organisms. So that puts people in a powerful position to start working across systems. DNA methylation, we already mentioned it's not possible to assume that information can be transferred between species. There might be a phylogenetic association, so if we know how DNA methylation works in one species of frog, we might have a good idea it might be relevant in another species of frog, but we still miss that kind of information. And there's always surprise. I mean, the thing is, there's always surprises. Like, it wasn't long ago we were talking about C. elegans not having DNA methylation. We're now talking about C. elegans having a different form of DNA methylation. So there is the fact that things like that can be found still just says that there are a lot of things that we don't know, and I could get into the don't know, the unknown unknowns and all that, but I think I'll avoid that. I think it's been overdone. And also, it's now not insurmountable to develop this knowledge de novo, from starting from nothing on a species to moving to quite a good knowledge of its genome and its methylome is not an insurmountable task. It depends on we discussed it was a lot easier for something with a very small genome than something like wheat with a massive genome, but it was still not an insurmountable task to do this. It's within the remit of an individual research group to develop this kind of information for a species of interest. So it's certainly an advanced position to where we were 10 years ago, that's for sure. And finally, these are our kind of key take-home messages about data gaps and research needs. And obviously, we could have put more money at the top, but we were very kind about that. We often came back to discussing terminology, and at various times, phrases like multi-generational, intergenerational, transgenerational, etc. were used loosely. We settled on intergenerational effects when we're talking about the transfer of effects between species as being a sort of universal term, really. So that was our suggestion, but we could do with getting the terminology right, particularly around multi-generational versus intergenerational and what we mean. Multi-generational being where generations are continuously exposed, intergenerational being exposure in one generation that has consequences for other unexposed generations. We needed also to have an important differentiation between knowing what was adverse and what was adaptive. That was important. We also a key question was about relative sensitivity. So we don't know anything yet. We don't know too much yet about the sensitivity compared to other endpoints. if we don't see changes on the epigenome until after the organism has died, then in ecotoxicology we don't really have to worry about spending a lot of time measuring things on the epigenome. But of course we don't think that will be the case at all. We also needed to know about reversibility and how long this memory was, how long it would last, etc. Whether true intergenerational effects occur at environmentally relevant concentrations. There are intergenerational effects published in the literature, quite a few of them. I actually sat and read the literature on this about 2 or 3 weeks ago to trying to get data together for some kind of meta-analysis. And there are quite a lot of studies out there, but they are generally at high levels. As I said, the radioecologists have this best, but then the radioecologists are doing their studies by taking a huge big block of cesium and putting Daphnia around the outside of it about 0.5 metre away. So whether that's a true environmental exposure is questionable. I think even they'd recognise that, but you know, we need to. You know, if you've got a nuclear power station, why not go for it? There are we know there are critical stages in development, but we don't know what they are. We don't know if they're consistent between different species, and that fundamental information is missing. Site specificity. We mentioned loci already. Which loci? How many loci? To what extent does changing those loci relate to an adverse effect? Also we're interested in the fact that the epigenome, it's not all bad, it's good. That's why the organisms have it. How do they facilitate adaptation of populations after exposure? Do they help? Do they hinder? If a population's exposed, do effects on the epigenome hinder? Are they impediment to recovery? Are they a facilitator to recovery? Or are they possibly both under different circumstances? So our key areas, we're looking at establishing adverse effects. Particularly, the key question was to know whether adverse effects of long-term exposure are greater than we would find from short-term toxicity tests. If they are, if we see adverse exposure and long-term effects causing changes in the epigenome that facilitate effects greater than we'd see from short-term tests, that's an important finding. That's very important for risk assessment. We needed to do that, we're going to need robust procedures to look at intergenerational effects and robust protocols. So we also need to think beyond standard phenotypes. Is just looking at births and deaths all we need to do, or do we understand subtle phenotypes? So things like behaviour, things like we talked about, behaviour in pollinators was mentioned. Really important adverse effects. Pollinators are known to have a very active epigenome. How's the link between chemical exposure and the effect on pollinator behaviour work? Does that can really affect populations. If bees can't find where to go to eat, then you don't have many bees. We also talked about things like how does changes to the epigenome of crop plants change things that we don't normally measure, not just yield, but also possibly allergen content and so on. We identified a need for consolidated data resources to allow structured databases and analysis and meta-analysis across chemicals, across taxa. Consider it, because we also recognise the important need of time. If we're going to develop understanding mode of action and AOP cascades, then we need to include time as well as dose in studies. And we also recognise we need to look at the costs of epigenetic change. So one of the things about adaptation is it can have benefits, but it can also have costs. And one of the examples of that we discuss is the earthworm study that Kevin mentioned in his talk, where we found epigenetic adaptation in one lineage. That if we take on an arsenic mine, if we take that population from an arsenic mine and move it onto a site where it's not polluted, that population can't survive. So there's a clear cost of that adaptation. Is that just a cost in the change? Is that a physiological change? Is it a physiological change that's been facilitated by changing the epigenome and so on? So there's a real need to consider cost in this adaptive phenotype. And with that, I'd like to abuse my position and and invite you to come to our meeting, a CTAC organised meeting on environmental omics and epigenetic sciences in Ghent in September, where all these topics will be discussed in glorious detail. And I'd also like to emphasise that for anybody at this meeting, we're going to extend the abstract deadline for 2 weeks, and we'd really like you to come and talk about all things omics and epigenetics, from archaeogenomics to regulatory epigenetics, and microbes and microbes, plants, animals, and people. So there we go.
Eric, thank you. You're also a salesman. Also, thank you again also for the members, participants of discussion group 4. So the paper is open for comments, questions. Please in the back.
Very nice presentation. I just want to ask you, how would you include neutral effects in your presentation or in your framework? Because it seems like that you're traveling between adverse and beneficial effects. But indeed, a lot of recent research has shown that the vast majority of evolutionary changes are indeed neutral.
Yeah.
And by neutral, I mean they have passed the detrimental. I mean, they are not detrimental, but they are not beneficial either.
Yeah, no, that's a really good point. I guess one way we'll include it in our presentation is by adding the word neutral in between where we mention adverse and beneficial. Yeah, but that's. I mean, it is a reasonable point. That is a reasonable point. Pete, do you have something to say?
I was going to say, one of the things we have to realise is, just like normal SNPs, there's a high degree of natural variation in epigenetic marks, and identifying when epigenetic marks, just like a SNP becomes an adverse disease carrying SNP or an advantageous adaptation, identifying that normal background, just like we've grasped this with normal mutations in the DNA sequence, epigenetic marks have to be viewed in the same way. So not all marks are going to be positive. Changes in epigenetic will have any phenotype. Some will have specific phenotypes. Obviously, we don't have the kind of rule set that we have with normal DNA changes, but that's the kind of understanding we really need.
Yeah, thank you.
Professor Gotti, you...
But in the very first slide, I mean, there was a sentence where you wrote epigenetic mechanisms can drive adaptive changes. And then in one of the last, I mean, you said that we need to differentiate, between adaptive and adaptation and adverse. Can you expand a little bit what you mean as adaptive?
Well, by adaptive we mean that when an organism is exposed to a stress,
No, just because the difference can influence very much the risk assessment.
Yeah.
Because if it's only, is an adaptation, it doesn't mean that it's a negative effect.
No, certainly not. But certainly not as long as that adaptation has no cost, which is why we mentioned at the end there about the cost of adaptation as well. So it's easy to look at adaptation, the upregulation of an enzyme system to detoxify a chemical, etc., as an adaptation. That's why we mentioned the need to make sure that we consider also the cost element to that adaptation as well.
Yeah, because adaptation is something similar to reversible.
Yeah.
For the risk assessment.
Yeah.
That's why
I I mean, my knowledge of environmental risk assessments at the moment is none of them do. Even though there is evidence of adaptation to some exposures, that's never taken into consideration in risk assessments that I know of at the moment. So, I know that the metals industry have been spending quite a lot of money looking at adaptation, with a view to suggesting, well, if species can adapt, why are we so worried? But that's certainly not accepted in the way that risk assessment is done for metals at the moment, for environmental risk assessment anyway.
Thank you. Eric in the back.
Yes, as I may have mentioned, risk assessment is not my field. But is there any consideration for environmental risk assessment and adaptation and potential evolutionary change for what we tend to observe in our model species is that after an environmental toxicant exposure, what we see is mostly just an increase in variability of phenotype. And you know, therefore more of them are diseased, but in a changing environment that may provide more for selection to act upon.
Yes.
And is that a thought about?
Well, firstly, I'd like to say that my business is a risk assessment either, so I'm certainly not going to speak for the way that risk assessment is done. As I said, at the moment, adaptation is not, to my knowledge, included in risk assessment as in the way that it is done at the moment. I agree with you that when you look at chemical stress, there's been the studies I've seen in ecotoxicology that have looked at whether chemicals increase or decrease genetic diversity tend to show them increasing diversity rather than decreasing. The ones that I know of, but that's still an area in development. I don't think enough is known about genetic diversity, let alone epigenetic diversity, to answer that question in a comprehensive manner, and because of that, it's certainly not part of current risk assessment. Well, to my knowledge, it's not part of current risk assessment, and I don't think the risk assessment is thinking of including adaptation in the near future as an assumption for certain chemicals, but not others.
Thank you. Are there other comments, questions? If not, Eric, thank you again, and thank you also for this announcement. May I propose something? It is not on the program. It's not in the program, but we need a little bit more time to finalize our take-home messages. Would it be possible to invite the 4 chairs of the discussion groups, if you have remarks, observations where we could react on, so that you say, okay, this is something what we, or what still has not been discussed, or what we could react on. So hopefully I'm clear, but it's of course depending on your volunteer, if you like to do it, to come forward or to sit where you are and give us some clues of course there was a lot of summary already presented, but perhaps the observations from the side of the chair that you say, okay, yes, please take note of it, because that is where we struggled with, or some other issues. Tina, you are...
I was struck by the remarkable consistency across the 4 discussion groups. I expected much more diversity, but in fact the findings were very consistent, and the most important, as someone who runs a research program and not a risk assessment enterprise, I walked away with a message that we need to make, if we are expecting to consider these effects, that we need to really invest in more clever study designs that will allow us to meaningfully probe these effects and understand if and how they should be incorporated into decision making. So the investment in the research, the investment in novel study designs, and then the other thing that almost every working group recommended is a revisiting of the risk assessment paradigm, and whether it can or even should be remodeled to absorb the extent to which complexity is being introduced as our understanding of biology is improved. And that was almost in every discussion group I walk away.
The other chairs who have to add something, what Tina is saying. Yeah. There's really also the feeling in your group that these 2 observations, we need more investment, we need more research between various disciplines.
Well, I can first echo what Tina said. That was my first impression when I saw this also. It's almost like we saw the same presentation given 4 times in different slices and different variations. Yeah, so remarkable consistency. What I'd like to add perhaps is that what we thought was really important is that studies are designed to really gauge would epigenetic effects create the most sensitive endpoint, in what situations would it, and how would we be able to pick it up? So actually design studies where we can dissect out epigenetic effects from all other effects, and then look at are they really the most sensitive? Are they at some point the most sensitive effects, and would we not pick them up with a normal study paradigm as with our standard tests that we normally are doing?
Diane, you?
Yes, hold on. Yep. Agree with much of what was said as well. I mean, one thing that was mentioned in the last one as well is the understanding that epigenetic changes are a natural part of biology, and a change doesn't necessarily mean that there is an increased risk. And so we need additional information before we'd be able to incorporate that into risk assessments. So, you know, additional research obviously required. Another thing that was pointed out on the animal side for the farm animals is that there's a lot of information missing, and also that certain changes are normal and transient through the life of an animal. And so you can have natural epigenetic changes that occur throughout the life. And then the difficulty in pinpointing what any changes in those normal changes mean. And we're not quite there yet. And with farm animals, you've got many different species, many different stages of life, and also different cells within the same body may have different epigenetic changes. And so keying on to what cells to look at as well, and at what stage of life would be important.
David, you asked us also yesterday very good questions about risk assessment, what to do. So was this also discussed in your group? So this more investment, but also what to do with the risk assessment paradigm, because I was involved in discussion group 2, and okay, it was to me also an observation, although I asked you the question and not should ask myself the question, but there was also some hesitation to say, okay, we have now a new point of departure.
Yeah, perhaps I should say that I'm just reacting now, but I think also in common with the other chairs, I agree. I think in many of the presentations, it's quite clear that this is a really important aspect of biology, epigenetics. We need to know about it and understand what it is, but then we've highlighted in general data gaps that need to be filled before we can apply it routinely in regulatory settings. I think that's what I've heard, and I think that's consistent with amongst the groups.
Sorry, I might add to that, because that was also addressed in our group, and it connects well to what Dave from Group 4 presented, that it is a natural process, and every test organism or every test material source has kind of a history of epigenetic change and adaptation. And I think it's not just for having new tests, but also using that information for assessing the test material and whatever we work with in risk assessments, if it's appropriate, if it's kind of different than other material, cell lines or whatever people work with. So it could be a tool for assessing the materials we work with better than we have it now.
Thank you. comments? You see, I don't try to find a solution, but we also had discussions, of course, not making a joke. A toxicologist should talk with epidemiologists, but also with medical people. As anybody, because that is what we are doing in this workup, to find ways to go. What could be done on the medical side of research to find the data, and what could be done on the animal side. So in principle, of course, I had a discussion yesterday also in our group to say, okay, we try to do it to go to the clinics and say, okay, what is your final outcome? What do you see? And then going back to what we see as markers. Of course, we are looking for markers. So, but you see, what is in my mind, you say, okay, it is a part of a natural process. So how could we do it? What is necessary? What could you advise EFSA to say, okay, at the clinical side, we should do this kind of well-designed experiments. So in order to you could call it validation or something like that. So is somebody who has an idea how to do this? Hopefully I'm clear what I'm trying to bring forward.
You mean besides put resources into research?
Yeah, of course, yeah. And okay.
I mean besides that.
If you say investment, then of course perhaps I say, okay, we should not come for the conclusion, but then I say, what kind of investment? What is then plausible to do, and where should we then put our money on? And
well, the thing I also want to say is that doing especially prospective population studies are very expensive, but there are a number of standing platforms that we could explore collaborations with to put some carefully posed questions and see if we could probe those studies and begin to at least use it to prioritize where the next steps should be in this area. So a little bit of collaboration and leverage.
Yeah, I was trying to raise the point, as we have discussed also in our group, which was Group 1, that I think that we can indeed learn a lot from data obtained by clinical scientists. So at least we can learn about the human epigenome at baseline levels and at disease levels from particular target organs. Where it also is the case that the methylome of target organs is maybe very specific. So translating our experimental data on the epigenome induced by toxicants to clinical data makes at least some sense, although as I have also said yesterday, we are comparing early events observed in our experimental models with late event observed in patients. But anyway, it would add to trying to generate relevance with respect to epigenomic changes induced by toxicants.
I was actually going to make the same point, more or less. I think the point is about clinical information is about existing disease. The markers, the epigenetic marks are about existing disease, whereas what we're after really are those causative events for the disease. I think there's still a lot to be learned from that engaging in that discussion, but I think that there is a big difference.
Okay, thank you.
Well, Huub, I think I'll repeat something that I said in our group, which is
the other ones for
of course.
Therefore, we try to.
I think yes. Of course, I agree with our chair that prospective epidemiological studies will be very difficult to conduct and expensive. And yes, there are platforms that you can apply to. But eventually, you could take the simple approach of looking, it's not my area, but looking at intervention studies in nutrition, because there you could actually relate nutritional interventions that are related to the C1 kind of metabolism, like folate, for example, supplementation of women, nutrition of women in childbearing age or infants and so on, and you can relate the biological outcomes with the level of methylation, with the type of intervention. That could give us at least a view as to where we should be looking if we go further into the toxicity testing.
Everybody agrees with this suggestion? Yes. Okay, thank you. Any other one who has an idea? how to use clinical data, and if it is possible to find the right markers. Because I'm a risk assessor, so perhaps I'm saying naive thing, but I'm interested always in markers to look at the behavior of compounds in organisms. If not, then I would propose that we have 5 minutes break, 10 minutes break. Andrea, how far are we? You are ready to give us some... Okay, okay. Wait. Jean-Michel.
Otherwise, I may have a suggestion. I mean, this was a very interesting meeting, and I mean, personally, I have learned a lot of things, but I have also learned that what we do not know is much bigger than what we do know. And what I think this might be a starting point to organize, to set a working group within EFSA on behalf of the scientific committee in order to try to implement or to see whether it's possible to implement some of the conclusions coming from the different discussion groups. Because I don't think that in 1 hour we can definitely conclude, but maybe we could start from this conclusion and work on that. I mean, I have suggested a working group within EFSA, but it could be anything else, in order to see whether it is possible in the near future to implement practically some of these conclusions.
Thank you for this good suggestions. As I also tried to tell you yesterday, within the scientific committee of EFSA, we have already 2 priorities. Looking at that, again, at epidemiological studies, what can we do with them? So that is already a topic. And then also of individual susceptibility, of course, then focus on the uncertainty factors. Are they, but yes, that might be a take-home message, and for sure that EFSA staff has noted it. Professor Ghali?
I think that it may be useful because I myself have some problem with the definition of this epigenetic change or marks or whatever. Because, I mean, I still wonder if the change is, I mean, the link of the methylation, the hypo, hyper, whatever, or the expression of the phenomenon. Because that is something, I mean, that is related to the receptors, protein. I mean, protein can bind the receptor, but if you are not the expression of this binding, I mean, the protein, it doesn't matter. That I mean, I'm thinking to the risk assessment. So, what is the weight of a change at macromolecules level if you will not measure an expression of that?
Thank you. Is this, you want to have the floor? Yeah, but it's not connected to previous. No. Okay. Jean-Michel.
Okay. Again, to let sometimes to be prepared to present the general conclusion, I will take the floor. What I mean is what you said yesterday and what you just say now. I am wondering whether there is a direct relationship between uncertainty and between reducing the uncertainty in the risk assessment and taking into account epigenetics. I would rather think that taking into account epigenetics would increase the uncertainty of the risk assessment. That's why I said...
that might be a misunderstanding.
So separate.
I only say it is one of the factors which deals with uncertainty. Yeah, of course, we don't know whether this is decreasing or increasing, so, but for sure we will look...
What I meant, again, I mean, just stop me if they are ready. What I meant is to think whether the current frame of risk assessment, as it is developed in the different panels of EFSA, really fits with those new concepts of integrating epigenetics. I am not sure. And I mean, the discussion we had within, at least within the discussion group number 2, really shows that it was very difficult, I mean, to integrate epigenetics factors and change within the classical frame of risk assessment or of tox studies.
Because, at least it was my understanding, but you are members of this discussion. My understanding was, is epigenetics something of a cascade of events leading to an effect that might have one of the steps? And of course, I'm honest to you, I'm a toxicologist, so I'm interested in the effect. And that was the discussion in our group. Are we talking at a direct cause for an effect? Or is the epigenetic some of a switch or a step which leads, of course, to this effect directly or non-directly, etc. So that is perhaps naive, perhaps, but it would be my summary. But I have to sleep and rethink all the wisdom you give us, and already starting giving you some kinds of thank givings, but I have to think about it. But that was a part of the discussion. Is it something within a cascade of events, or is it already something but pinpoints directly to a liver lesion, etc., etc.?
May I make a joke, Mr. Chairman? Concerning the uncertainty, because in the opinions, in the average opinion, at the end you have usually you have plus minus whatever, you know, a scheme of on the uncertainties. But in this case, I think that is very easy, because if you sum up all the may and might present in these slides, you understand how the uncertainty is.
Sure, we are still keeping the current paradigm, but of course, this is our job to take all this wisdom on board. And please, in the back.
So I understand it's not really the point of this meeting, but I think we need to remember and perhaps take a note that we are now on the edge of having technologies to intentionally change the epigenome of plants or farmed animals. And so there will be a whole area for risk assessment and regulation of these new kind of organisms. So it's not part of this meeting, but just to take a note that epigenetics and risk assessment has also this other avenue which we'll have to walk down at some point.
Thank you for bringing us back to reality. So yes, of course, perhaps it was too narrow discussion between risk assessors, but thank you, of course. There are a lot of other possibilities. So thank you for having this intermediate discussion. So Sandra, may I invite you to give us some take-home messages? And also you, thank you for the willingness to do this in a too short time, but the floor is yours.
Thank you. This was not an easy task, but I did my best, so let's hope this is going to work. And yes. So now we come to the close of what has been, by all accounts, a very successful and productive scientific colloquium focused on a very interesting and challenging topic, epigenetics. And we all agree that epigenetics is one of the most rapidly expanding fields in biology. Just by looking in PubMed, you can find that in 2010, 940 approximately papers were published, you know, if you search for epigenetics, and after 5 years, there were 3,500. So the interest is very high, and we understand why. So what about epigenetics and risk assessment? Where do we stand? Let me recall, before we go into the general conclusion, the main general objective of these very intensive days of presentation and debates. This is the No.
You have to do this.
It's on. It's on. Oh, sorry.
No, you've got the first slide up. Thank
Okay,
you.
so it works. Good. So the general objective that we all had working in different working groups were to discuss whether epigenomic data should be integrated into risk assessment and whether this would make a meaningful contribution. And then we had to try to address molecular mechanism, methods to investigate epigenetic effects in vitro and in vivo, and the use of epigenetic biomarkers, possibly, and to identify the existing data gaps and the research that is needed. And, oh no, sorry. No, I want to have another one. Yes, this one. We had excellent speakers that gave an overview about relevant topics. And here you have the summary of the titles of the main lectures that we had. So we got to know more about the environmental and nutritional effect on epigenetic regulation, the epigenomic molecular mechanism of chemical injuries, the role of epigenetic in toxicology risk assessment with special focus on furine in food, and epigenetic and risk assessment of farmed animals for food production. And the final lecture was on the impact of epigenetic in environmental risk assessment. So already during the presentation and afterwards, some points for discussion were identified. And I try to make a sort of short summary because, of course, it was more. So the DNA methylation changes that we observed are caused by the environmental cues, or rather do these changes reflect developmental or tissue alterations? Why are certain gene loci more highly susceptible than others? Are the epigenomic alteration of this genomic loci consistent and big enough to be considered biomarkers? And which epigenetic markers should be considered? So you will see that these questions come back when we present, I try to summarize the discussion from the working group. More points. More points were also related to the differentiation between protective adaptive epigenetic changes and adversity, the degree and determinants of reversibility, and the complexity of multiple DNA and histone modification. Another point was related to the dependency of potential transgenerational effects on dose exposure, critical stage of development, and species. And an important aspect that came up was the need to check the F3 generation to claim transgenerational effects, something that sometimes we forget. So the discussion then moved to the different groups, and you have heard a very comprehensive summary from the rapporteur of each group. So my task was to identify points that were shared by the groups, because of course you can go back to the single presentation to get more information about a specific point in the discussion. And the take-home message and questions that I identified are the following. We all agree that we have limited data about epigenetic changes induced by chemicals. So there is no doubt that this is the current situation. Instead, there is more knowledge available on specific epigenetic modification occurring in pathological conditions. And so the general agreement, it seems that we have to take advantage of the progress made in the clinical fields and in epidemiology to see how this knowledge available can be used to advance the risk assessment. This is a general point that all groups took up in their discussion, I would say. Questions, also quite common. What do epigenetic modification mean? And it seems to be really important for most of us taking part in the discussion that we anchor to phenotype or to gene expression modification to give some relevance to the epigenetic modification. Now, the question is, what is the size of the effect that we need to worry about? And this is an open question. There is no answer right now. Another relevant question that you have addressed, that we all have addressed, is how do we study epigenetic modifications? And it's very clear that we have to use different types of models. And we can select in vitro or in vivo models, which are relevant for the targets that we want to study. So target-specific models would be first choice, at least from the discussion we had. And another important point is that different types of organisms should be considered, and this is a relevant point for ecotoxicology. An important note that seems to be also shared by several of us is that the current toxicology studies may in principle allow to monitor adverse effects resulting from epigenetic changes. So we have the possibility to identify these changes by doing the tox studies that are currently in place. But one of the potential weaknesses is that we may not identify transgenerational effects. So this is a big hole that we have in the study. We also have identified some gaps, of course many, but let's try to see some of them. And these gaps are relevant for both toxicology and ecotoxicology, but also farming. We need to better understand epigenomic, and special mention was for mitochondrial DNA by some of the group. We need also to better understand the cause effects between epigenetics and outcome. And then we need to develop and validate testing models and cost-effective platforms. Again, the need of more translational science seems and appears to be necessary in all fields that we have considered during these days at the meeting. So, where do we stand? We can say that oops. It's gone. Oh, now it's gone. Well, it doesn't matter because the summary is over, so I can continue without slides. It seems to me at the end of this day that we have identified what we know and what we don't know. And I think this is a big achievement, because once you know what needs to be done in the future, then of course you have identified the way on how to proceed. And I think this the need of the cooperation, the collaboration with the clinical side, with epidemiology, and other aspects that can be considered by collaborating with colleagues from different disciplines is also a plus in the strategy that we should have for improving risk assessment. So at this point, I'd like to thank EFSA and EFSA staff for the excellent organization of this meeting in such a fantastic location. And I want to give a special thanks to the speakers and the chairs, the rapporteurs, and that have helped me quite a lot in putting together this presentation, since I got the take-home message from the different groups a few minutes ago, basically. And of course, a big thanks to the participants, because thanks to their contribution, this colloquium can be considered a big success. Thank you for your attention.
Thank you, Sandra. We finished a little bit earlier, but I think what had to said has been said. So also on behalf of the organizing committee, but it was already done by Sandra. Sandra, don't go away, please. Take the seat. Because this sitting there. So thank you again also on behalf of, as Sandra said, organizing committee. I think you give us a lot of wisdom, a lot of results where we have to chew on within EFSA context. So as we said, it will be some kind of booklet. I have only a few things to say. First of all, I wish you all a safe trip home. The meeting documents will be available shortly at the EFSA website. And hopefully that I was clear that there's already a PDF file available of the reports of the 4 discussion groups. If you need a certificate of attendance, please approach the Secretariat. And if you need some kind of a taxi booking or something other transportation, we also would be glad to assist you. But thank you. Thank you for your honesty, your transparency. Really, as a simple toxicologist, I learned a lot, so I'm very tired, and hopefully that you're also satisfied what we organized in the context of EFSA. So thank you.
Outside the secretary. Yeah.